Okay, everyone knows that all the important events in the history of life on Earth occurred in the ocean. Life originates there, we get the first eukaryotes there, we get the first animals there, blah, blah, blah. The only important thing that happens in the first 90% of Earth history on land is when it gets covered in ice and a load of nutrients get washed into the ocean. That's the story. However, this is beginning to be challenged, and people are starting to think about what actually lived on the planet's surface early in Earth history and how this affected the Earth system. And there's actually been a slow but growing revolution in Earth sciences where people are starting to think that many of the major events in the history of life may have actually happened on the planet's surface rather than in the oceans. And that's what I'll discuss today in a very terrestrial biased talk. However, there are a couple of caveats because it is difficult to study early life in the terrestrial environment. Essentially, it's difficult to recognise when you have a terrestrial environment because you're based on sedimentological and non-evidence, essentially. So differentiating between a marine and terrestrial Archean environment is not easy. Also, once you get a terrestrial environment, it's difficult to preserve organisms in it, and many of the things we'd expect to find there are difficult to preserve anyway. So the fossil record's pretty poor. And when we start to look at modern organisms, the molecular clock analyses are often very inconsistent when we go very deep back in time at these early divergences. Nonetheless, if you go to most textbooks, they'll accept that there was a pre-land plant terrestrial biota, and it's usually considered to have been microbial, living in ponds, around lakes, freshwater environments, maybe as soil crusts, or even within rocks as endoliths, the sort of things we get in Antarctica today, UVB protected and so forth. But we have very little actual evidence for what these organisms were and indeed what their ecology was. When we do want to study them, we can rely on two things, obviously. We can look at the rock fossil record and we can look at living organisms. And I'll just briefly run through the different types of rock fossil record we have, and then I'll go through a history of the invasion of the land, if you like, right from its beginnings up to the origin of land plants. We'll then finish up by looking at the living organisms. So there's some very interesting work coming out on this based on the sort of ancestral state reconstructions we've just heard about. So, fossils. We get the actual fossils when we go onto the land. We can dissolve up our rocks and we get all sorts of palaeomorphs out of there. We can take thin sections in the rare cases where we get phosphate nodules and the like, and we get promineralizations. We also obviously get stromatolites and various microbial sedimentary structures, and these occur both non-marine, marine environments. Then we can look at isotopes and biomarkers, just as we can elsewhere, and these also preserve in soils. So a lot of the work I'll be talking about is done on soils, early soils on the land. And the isotope work, carbon-13, carbon-12, is pretty well accepted. Biomarkers less so. Awful lot of work's gone into this, but there's always problems of contamination, and there's problems of, where do these biomarkers actually come from? How good are they at identifying different organisms? There's a whole group of people that also look at different weathering signals, both in the marine environment and also on the land, the type of things that should end up in the ocean that give you clues as to how the land is being weathered and therefore whether you've got a biota on the land when it appears. And these kind of things by Nath and Kennedy, where they look at the late Precambrian greening of the earth, is a typical example of this. Fourthly, we actually get paleosols, paleoclast, laterites, old earth surfaces. Some classic examples from Greg Rotalic. This is one of the Torridonian soils. Here you've got the unconformity surface. He looks at this and will take you through the soil profile and so on. Right, so this is my version of the diagram you should never show, where everything is controversial, starting with the accretion of the planet and moving up through. There's the oxygenation state. Maybe we get ozone layer here here's the different glaciations, and so on. The important bits are the green bits, and this is where we start to get evidence for terrestrial life on the planet's surface. So we actually start way back down here with the Fortescue group and the light several sub-Transvaal group, Palaeontological and so on. We're not as far back as the earliest evidence for life, but we begin here, and right the way through here we've got a whole series of soils we can study, and then we actually start to find lake deposits where the sedimentology pretty reliably tells us they're freshwater lakes or rivers. We go through this series up here, all the way up to the origin of the land plants about here. And I'll run through these in order from the oldest up, and we'll just think about the different types of evidence for life on land. So if you go to the Fortescue group way back, there's different lines of evidence here for life on land. There's paleosols in there that people have looked at, and when you study the isotopic ratios there, there's good evidence that there's a biogenicity there, there's life that's been variously interpreted, and certainly methanogens, there's strong evidence in the biomarker record for methanogens and the like. Is that me? No. We could also, in there, we find... Is that working still? Yeah, good. We find evidence for oxygenic photosynthesis from stromatolites that Buick has described. Part of the problem here is people have questioned whether these are actually terrestrial or marine. So although you definitely get these paleosols, is it marine strata that overlies it that's giving you the signal? And also, are these kind of stromatolites actually in marine environments, non-marine, tidal, and so on? So it's always been slight questions hovering over these. Same when we go to the 2.7 Ventosdorf supergroup. You get these stromatolites in ooids that are clearly produced by microbial structures, but are they marine or non-marine? You get raindrops in these sequences, you get soils in these sequences, but when you actually get this first evidence, it's always very difficult to identify a true non-marine environment. Moving up, we get the 2.7, 2.6 paleosols underlying the Transvaal supergroup, and geochemical evidence here is pretty good, suggested by Watanabe et al., suggesting we've got life on land at this early stage. So there's something out there. And there's a whole series of soils that take us up through the great oxygenation event that have generally been analysed in order to give us an idea of the O2 levels in the oxygen. But obviously, as a byproduct of this, there must be life on land there based on the signal we're seeing, and there are all sorts of cyanobacterial signals. I won't go through all of those different soils. There's a whole series of them. Eventually, you start to get organisms that are clearly terrestrial. And this started off with Hoffman's description of material from the Belcher Island Formation, and it was reinterpreted by Golubic and Campbell to suggest that they were truly cyanobacterial and produced in these kind of intertidal zones. And they had this hypothesis of how you could therefore get a terrestrial invasion of cyanobacteria from the ocean out onto the land, and with cyanobacteria leading the way. Eventually we find something that I've actually worked on, and this is the 1.2 GA star group up in the Torridonian of Scotland. There it is, but it lies unconformably below the Torridonian, and it's a massive red bed sequence. It's just like the old red sandstone really, but much older. Occasionally you find these lake deposits in there, and some of them have these MIS layers that you can section and are almost certainly created by microbial mats. You also get evidence of subaerial exposure. There's raindrops, there are desiccation cracks, and so on. And when you dissolve up the lake deposits, what you find in there are organisms. Actually, a long time ago, Cloud and Germs described these. They're not very well preserved. It's been heated to a huge extent. But when we re-dissolve them up, you get a very simple biota of microbial structures, certain sort of small cyanobacterial structures, but then larger spheromorphs, and some of these are up to about 50, 60 microns, and kind of moving into what we generally consider to be eukaryotic range. Soils, again, about the same age from the Mescal limestone in Arizona. These have actually got these superb cyanobacteria preserved in these interclassed concretions that have been preserved in there. And when you section them, Horodisky and Nauth describe these beautiful cyanobacteria that were either washed in or a part of a mat, a microbial mat, that are actually living on the soil horizon. Again, we look at these things, we have to interpret them as terrestrial first of all. We then have to try and work out what the organisms actually represent. These are pretty good in terms of cyanobacterial. By 1.1, we're moving into the Oronto group in the Michigan, USA. We're looking at a huge sequence of terrestrial deposits that were deposited in the mid-continent, starting off with the Copper Harbor conglomerate, go through the Nonsuch shale and up into the Frieda sandstone. And these for a long time have been known to be non-marine, classic terrestrial sequences, and people have investigated them for different lines of evidence for terrestrial life. Starting off way back with Barkhorn, who looked at the biomarkers in there. A lot of this work has been slightly discredited now, but he found biomarkers he thought were, or they found biomarkers they thought suggested cyanobacteria and various terrestrial organisms. Moore dissolved them up, found some paleontological remains. Some of those may have been contaminants, certainly some of them are, but some seem pretty genuine. And Elmore had a look at the Copper Harbor and found these beautiful stromatolites, which are in place and seem to occur laterally as you move from the lake system out. So they're kind of lateral equivalent of the Nonsuch. When you dissolve up the Nonsuch shell, you actually get your first really good palynological assemblage. What we have in there, this is work I've done with Paul Strother. A lot of the initial work actually works at the copper mines, where hydrothermal activities kind of ruin the paleomorphs. They're not very good. When you look at the boreholes that are further away from the mine, you move away from the hydrothermal activity and you start to get beautifully preserved paleomorphs, and this is the natural colour. They've just been led around on the mid-continent for 1.2 billion years. In there, you get the cyanobacterial remains and the other microbial remains, these small little structures, generally 1, 2, 3 microns in size, and you get the filaments. Also, you get this much larger biota of all sorts of weird and wonderful shapes and sizes, spheromorphs, forms with internal bodies, forms with these spots, forms that clearly open up to release, assist at releasing something, and you get groups of these together. You also get these strange flask-shaped structures. And the Nonsuch is really incredibly diverse. It's as diverse as anything you'll find in the marine environment from this age. Interpreting it is interesting. What do these organisms represent? Other than saying prokaryote, eukaryote, it's very difficult. There's the odd organism we look at, we think we may have euglenoids in there, and so on. But actually placing these is not easy, and that's one of the problems. Particularly when we go to look, therefore, at the ecology. Are these living in the lakes? Almost certainly. Or do they also represent material from the land that's been flushed in, and so on? Whatever, we appear to have a very diverse lake environment by 1.1 in the Nonsuch. Interestingly, a lot of these little cyanobacterial bacterial remains tend to congregate like they do in modern organisms to produce these kind of large structures which then can become planktonic. We're not sure if this is part of a mat that's just broken away and these could be bubbles from it and so on, or if they're using this structure that modern cyanobacteria do, where they kind of congregate and then live a planktonic existence. There's all sorts of interesting things to look at when you go to the Nonsuch in terms of organisms and what they actually are, what they're doing. Likewise, when you go to the 1GA Torridon group up in Scotland, we look at Stewart's sort of classic reconstruction. It occurs in between mid-continent at the time, and it's just masses of rivers, these kind of braided rivers, dumping this 7 kilometre thick red bed sequence. predominantly conglomerates, sandstones, but then you get the lake deposits in there and the old fluvial deposits, which are unreduced, and you can dissolve these up and look at the Palaeontology. Pretty well dated to just less than 1 billion years old. There you have the sleep group, equivalent to the basal of the Torridon, and there's that huge thickness of the Torridonian. And we've got microfossils virtually throughout this group. Every different formation is yielded. These microfossils, interestingly, were known for an awful long time, actually. Till described them in 1907 from phosphate nodules, and that was one of the first descriptions of Precambrian life, and kind of just is hidden away in the literature in the back of the Geo Survey publications until it was rediscovered. Many people have looked at it. Downey, Normova, Zhang did some interesting work on this material. But it was all bits and pieces. So eventually I got together with Paul Strother, Martin Brazier and co, and Lila Batterson, and we sort of reanalysed this whole sequence in terms of the palynology, sedimentology, and also these phosphate nodules, which beautifully preserve these microfossils. What you see in the lake deposits is quite extensive lakes. You can see the ripple laminations, but you get large scale drying out structures, desiccation structures all over these surfaces. In between them, you can see the microbial mats, these mist structures, and you actually get raindrops in there as well. It's a really good terrestrial environment. We have no problem with describing this as terrestrial, and when we dissolve it up, we find beautiful microfossils in there. Looking at the mats, what you find are relic bits of mats run through with what presumably are cyanobacterial sheaths. You also find bits run through with these microbes, similar to modern Euhalithece, the sort of things you find in the Sahara and these little ponds that dry out. And they're the classic sort of size you'd expect for microbial life. But like the non-such, there's also this much larger element in there of these big microfossils. And these go up to 0.5 millimetre in size. All sorts of different spheromorph types, variously folded. Many of them have these opening structures where they've encysted. Often they have the spot. Some of these we have attached to tubes, and others appear to be more internal structures. So they're not necessarily where the sort of cell content is has basically shriveled up. Some of them appear to be actually on the surface and where they occurred at the end of a tube, or a tube grew out of it. I'll show you some of that later. But these occur in clusters, and there's all sorts of weird and wonderful fossils that are very difficult to interpret. What are they? What's living on the land here? What did it live? What was the ecology of these things? Are they actually living in the lakes, rivers, on the land, being flushed in? Currently, we think our ideas are that they are living in the lake, but they're predominantly benthic. And so we don't think we've got a kind of lake planktonic community yet. There's certainly nothing with spines on it, only these very large cysts. Bearing in mind, of course, that all we have is a cyst record. Large tuaria types, that's obviously half of one of these things. That's up 0.5 millimetre in size. So there's big old organisms around our Torridonian lakes. Weird flask-shaped structures. These are quite common. They always have this curve on them, and some appear to have a top. We've even got them where you've got lots of these small spheromorphs coming out of them. Again, very, very difficult to interpret what they may be. And some of these palomorphs have these bispiral ornaments that we think are euglenoids, certainly euglenoid-like. The key to this will be trying to actually get enough of them to section and prove the structure is euglenoid. And all sorts of scraps, bits and pieces, some of them have these kind of edges on them that are rather mysterious. What do they represent? And finally, we get these large thalloid organisms. They're either bipolar or they sort of tripolar thalloid organisms. That's 100 microns. So these are well millimetres in size, and they're falling out of some of the preparations. And they have a very characteristic kind of shape to them, but we don't know what they are. What does help is the work that Lila Batterson did on the phosphate nodules you get throughout these lake deposits in the Torridonian, and she sectioned them, and you get these beautifully preserved, three-dimensionally preserved fossils, which of course we can then compare the palaeontological which have been flattened. And again, you see here we have these are internal bodies, and you can see them dividing and so on. And also in there you get bits of mat preserved, so you get these rather large chunks run through with the different tubular structures, the sheaths and so on. Right, so that's a Torridonian. When you move up from the Torridonian, you go back into the soil record. Horodician now also described 0.8 million year older, really nice cyanobacterial remains. And then you get on to my good friend Greg Ricatalic, who much of the soil work has come from Greg, who's been spent his life interpreting the soils, but sadly then went on to interpret these and upset a lot of you by calling them lichens, I believe. Greg seems to see soils everywhere, and he now sees them in the Ediacaran sequences, and every soil he looks at, he now sees Ediacaran fossils, which he has, to upsetting me, been describing as slime moulds and fairy rings. Now, I've got nothing wrong with that, but I just, I can't see the structures and interpret them as lichens and slime moulds. They're very difficult to preserve. We generally only get them in amber and so forth. And in 2.3 billion year old soils, he now sees these structures, and I just cannot interpret them as biotic, let alone those structures. I don't say he's away with the fairies, but this lot are. These are my good friends Paul Strother and Willie Taylor, looking at the palynology of the Cambrian. They later found these non-marine palaeonomorphs in the Cambrian, Middle Cambrian record, which they initially interpreted as the spores of land plants. I don't think they are. I think they're actually from green algal remains and the material that's actually living on the land rather than true land plants. And I think they've more or less come to this conclusion. There's some great work they did with Linda Graham, where they looked at Coleochaete, which is one of these green algals. It's meant to be the sister or potential sister group of land plants, lives in freshwater ponds and so on. When that pond dries out, it takes on a totally different growth form, grows in these single cells with very, very thick walls. And when you section these, they're very similar to those Cambrian fossils we saw previously, and show many of the structures. I think what we're seeing here is the terrestrial green algal remains that are certainly non-marine, of plants that were similar to land plants, or maybe precursors of land plants. But that's what I believe they are, rather than the early evidence for life on land. Right, then eventually we keep going on up, and we find evidence for fungi, which is always interesting, because I did a whole series of experiments on modern ecosystems where we palynologically process them, and you see what comes out. And what we find is they're all run through with fungal filaments, something we don't find in the fossil record until much later. So this idea that fungi came on the land early, I think, is incorrect. I believe they came on with the land plants much later. The fungal record of life on land is interesting, and we'll hear more about it during the course of this conference, but I certainly don't think it's early or pre-land plants. Right, so eventually we find good evidence for life on land, and in terms of the spore record, we get the odd scrap of these Ordovician sporangial contents. I put that into Upset Diane. It's a Paleo bryophyte, just to show that what we're finding here are the sporangial contents and none of the rest of the fossil. So we really don't know what they are unless we believe the phylogenies. But it's good evidence there's life on land. This comes in at the mid-Ordovician. And so I'll stop there in terms of the fossil record. Now I'll just briefly turn to the living organisms, what we can do with living organisms, because there's been a huge progress in this area recently, basically based on phylogenetic analysis and construction of evolutionary trees, coupled with the molecular clock analyses, and then ancestral state reconstruction. And a lot of this or the interesting part to me was started by Battistuzzi with Blair Hedges and the like, when they did this very early phylogenetic analysis of the cyanobacteria, or bacteria as a whole actually, eventually, and then applied the molecular clock studies to it. And they recognised that the cyanobacteria are fundamentally a terrestrial group. And they actually went as far as to call them the Terrabacteria, this large group here, and said that the early divergent, or many of the early divergent bacteria, are actually terrestrial forms, and they therefore suggested that these appeared on land very early. Their clocks estimated at about 3.5 billion years, so sort of soon at the same time as we got our earliest fossil evidence for life on land. And that life, many of the important events, including oxygenic photosynthesis, may therefore have evolved on land. Subsequently, Karim Blanc and workers undertook similar analysis of the cyanobacteria and came to the same conclusions, that they're fundamentally a terrestrial group, and they plotted on different character states. They also did a molecular clock analysis which suggested, again, early origins of cyanobacteria on the land. Their character state reconstructions suggested they would have been small-celled and unable, interestingly, to form cyanobacterial mats. And it's only when she says that they later invade the marine environment at about 2.4, about the time of the GOE, that they can actually start to produce mats in the oceans. And kind of all these evidence from these living organisms is beginning to pose some really quite interesting questions as to where did these major events in the history of life on Earth happen? Was it on the land? Was it in the ocean? And what were the environmental effects of this? Green's also done the same type of study, but on eukaryotes, and they found the same thing, that the basal eukaryotes, she believes, are essentially terrestrial, and it's only subsequently 1.2 or so that they go into the oceans after they have diversified, or that many of the early diversifications have occurred on the land. So, in conclusion, I think there's evidence from diverse sources that life appeared on the land early in Earth history. We have to go away from this notion that we have this barren wilderness, it's totally sterile on the land until plants appear there in the Ordovician. This evidence comes from pretty good fossil evidence now, certainly looking at the soils and then later these decent lake deposits. But it's also coming from a study of living organisms, which is suggesting that, not all of this happens in the oceans, and many of these groups may fundamentally be terrestrial that later moved into our oceans. So these predictions based on ancestral state reconstruction suggest that some of the major events occur in a terrestrial environment. Of course, this means that we have a terrestrial carbon cycle, so we have two carbon cycles at the moment, the oceanic and the terrestrial, and it suggests we have an early terrestrial carbon cycle, which again we will then have to factor into when we reconstruct environments on land, geocycling and geochemical cycling and so on. However, we must recall that these early terrestrial ecosystems would have been pretty much microbial soils, things living in lakes and rivers, and their effects on the carbon cycle will be not nearly as much as when land plants appear and start to change the world forever. Thank you.